This is Lab 6 Passive Transport by Salim Shah, Nick Lee, and Neil Patel. This is just a basic organization of our presentation. We have this video, which is gonna include the introduction to membranes, which is the introduction for the overall experiment, and 4 other videos which include their separate introduction and conclusions. So, an introduction to membranes. Membranes play an essential role in cells. They are seen as the phospholipid bilayer and the fluid mosaic model. One function of membranes is to maintain cell shape. They also act as a barrier by separating the cytoplasm from the extracellular fluid. The phospholipid bilayer is selectively permeable, and hence it regulates the flow of ions, gases, and macromolecules from entering the cell and also exiting the cell. There are many reasons why this occurs, one of which being polarity. The phospholipid bilayer is arranged in 2 leaflets, which each leaflet is composed of the polar heads and the nonpolar lipid tails. As the bilayer is constructed, the nonpolar tails are grouped together on the inside or the middle, while the polar heads are on the outer edges of the phospholipid bilayer. And hence, things cannot pass directly through the bilayer because polar molecules are not going to want to go through and be with the nonpolar lipid tails in the middle. So here you can see right here you can see the nonpolar lipid tails, and this is one leaflet, this is the other leaflet, and you can see the lipid tails are arranged together on the inside or the middle. There are other parts to a phospholipid bilayer, including proteins, as you can see here. Now, these proteins act as helpers or transport proteins in the form of pumps or channel proteins to either participate in facilitated diffusion or active transport. Here you can see a carbohydrate marker. There's cholesterol here, which is another main part of the phospholipid bilayer. Another carbohydrate here, and these are the phospholipids again. Phospholipids are the most abundant component of the bilayer. A phospholipid is this molecule right here. This whole thing is a phospholipid. It's an amphipathic molecule, which means there's both a polar region and a non-polar region. Now here, sorry, one second. Here you can see the polar head, and here is the non-polar tails. Now the non-polar tails are lipids, they're actually hydrocarbons, while the polar heads contain a phosphate group. There are proteins in the cell, in the phospholipid bilayer, which function as transporters, such as channel proteins, protein pumps, and symporters. They also act as receptors, and they also play a role in recognition when you look at glycoproteins, which would have a little carbohydrate sticking out and identifying a certain cell. There's cholesterol in a phospholipid bilayer. These are located with the nonpolar lipid tails, and cholesterols are lipids, And the function of cholesterol in the bilayer is to maintain membrane fluidity based on the temperature of the environment. There are also carbohydrates in the cell, as we talked about before. These are used for cell recognition, and glycosylation is the process which adds the carbohydrate chain to the protein, as we see here is attached. The forms of carbohydrates are glycoproteins right here and glycolipids right here, as the carbohydrate chain here is attached to the phosphate head.